Food preservation is defined as the process of techniques undertaken to maintain food characteristics. Since foods are organic substances originated from plants or animals with various nutrients, they undergo spoilage due to microbial, chemical, or physical factors. Therefore, the main objective of food preservation is to increase the food's shelf life, retaining its original characteristics for a longer period of time. This presentation will address the advancement of food preservation from existing industrial methods to novel technologies with their mechanisms, application conditions, advantages, and challenges. In addition, health and environmental considerations will be examined to suggest safety guidelines for industrial applications.
1. Current methods and challenges. This section will cover predictive microbiology, microorganism growth factors, thermal processing for food preservation. Predictive microbiology is a scientific method used to assess microbial population. Usually, a study of food matrix is conducted to obtain the physiochemical characteristic needed for defining baselines. Then, targeted experiments can be designed for the collection of microbial growth data, which will be later used for initializing the primary model after fitting them with nonlinear regression to generate a prediction of growth population. The secondary model takes more environmental variables into account to improve the accuracy of each prediction. With such, complex polynomial, probabilistics, or artificial neural network are often utilized within the secondary modeling stage for more complicated calculation. The progress of food preservation has always evolved around the factors that contribute to microorganism growth. Three of the most important factors are outlined here. First, water is indispensable for the growth of microorganisms. The water requirement for microbial growth is often referred to as water activity, characterized by the ratio of vapor pressure found within the food and pure water. Under normal circumstances, a water activity above 0.8 is optimal for the reproduction of most microorganisms. Similarly for pH, each microorganism has a preferred pH range that allows for maximum rate of growth. The range varies differently depending on the type of organism. A common mechanism for pH manipulation involves artificially moving pH into an unfavored zone, thus forcing the cells to balance the hydronium level by consuming energy until they die of exhaustion. Aside from pH, the food content is also important when it comes to microbial growth. Most organisms siphon power through the consumption of simple carbohydrates and amino acids as their excellent source of nitrogen and carbon, which are essential to their very existence. Thermal processing is primarily utilized for food preservation due to its effectiveness of inhibiting microbial growth. The most common practices adopted, such as pasteurization, sterilization, and evaporation, all depend on the conductive and convective heat transfer, as the inactivation takes place through the irreversible denaturation of enzyme membranes. Both crucial to microbial survival, therefore cellular death is often inflicted as a result. For instance, milk can be pasteurized at a low temperature for a longer time, known as the LTLT treatment, which takes place at 63 degrees Celsius for 30 minutes, or certain HTST treatment at 90 degrees Celsius for 0.5 seconds. To evaluate thermal processing, three most important equations are showcased here, where time associated with a 90% reduction in microbial population is defined as a decimal reduction time d. The temperature differences required for the thermal inactivation curve to reflect the reduction of log 10 amount is often referred to as Z, and the process lethality often known as F.
Extensive studies have been focusing on developing preservatives that are non-toxic but able to extend shelf life as effective as traditional chemicals. As a solution, bacteriocins have gained increasing attention to naturally control the shelf life and the safety of foods. These secret chemicals that inhibit or destroy unwanted organisms in food through their metabolic processes. There are currently two major methods of applying bacteriocins, including encapsulation and inoculation of lactic acid bacteria with food. However, limitations still exist as the maximum limits vary between countries, and since niacin is the only bacteriocin licensed as an approved food preservative internationally, further studies are required to discover more types of microorganisms. Appropriate storage method is a key for safe and perishable food packaging. Modified atmosphere technique replaces normal air from the package with gas to minimize food's metabolic activity. Such examples are carbon dioxide, oxygen, and nitrogen, and they can slow down the growth of aerobic microorganisms. A similar technique is called vacuum packaging, where no additional gases are inserted after vacuuming the headspace of the storage area. Another popular storage method is called active packaging, where specialized materials actively interact with food and the environment to maintain or improve the condition of the food. Active absorbers remove unwanted components such as oxygen, carbon dioxide, ethylene, moisture, and aroma, while active releasers emit beneficial active materials such as antimicrobes, antioxidants, aroma, and carbon dioxide. The main applications have focused on delaying oxidation and controlling aroma moisture level or microbial growth rate. Recent coating technology is aimed to apply a layer onto the food surface to protect food items from environment. Previously, coating technology on the macro scale has been developed and implemented on bulk food item surface. Recently, microscale coating technology, also referred to as nanoencapsulation, has been studied to coat food ingredients on the microscale. A major benefit for applying encapsulation technique is that sensitive materials could avoid exposure to changes in environment during processing stages. It is also able to mask undesirable odor, flavor, or taste. In addition, studies have shown the ability of controlled release of encapsulated materials over a long period of time.
3. New developments in preservation techniques. Osmotic dehydration. Dehydration is the most efficient method for food preservation. Osmotic dehydration is emerging as an alternative drying method which aims for partial removal of water. The drying force for osmotic dehydration is osmotic pressure gradient between the food cells and the surrounding solution. Therefore, water molecules will migrate from food cells to solution across a cell membrane with the benefit of lower energy requirements and less substrate damage. For instance, osmotic dehydration is often conducted with food being immersed in an aqueous solutions that contain certain osmotic agents like sodium chloride, glucose, or phosphate. Ozone treatment. Ozone is an oxygen allotrope that has a high reactivity, making it an ideal candidate for food disinfectant. As ozone can undergo spontaneous decomposition into oxygen, it is preferred over chlorinated chemicals to avoid chlorine-based residue. To demonstrate, ozone has a strong oxidation potential of 2.07 millivolts, which surpasses permanganate and chlorine dioxide at 1.68 millivolts and 1.57 millivolts respectively. Ozone, with its high oxidation potential, has the capability to react with microorganisms through reactive free radicals, while destroying cell membranes and intracellular constituents that eventually led to the inhibition of microbial growth. Pulse electric field. As thermal processing discussed above is more prone to affect food textures, pulsed electric field emerged as another alternative for food preservation. In general, pulsed electricity is charged through the food to overload microorganisms within and inflict irreversible damage to their cells. Therefore, food is usually connected between two electrodes with a modulator generating pulsed electric field that allows high voltage in the range of 15 to 80 kilovolts to pass through. Each pulse is only applied for micro to nanoseconds as the large surge in power could generate excessive heat that changes the texture and structure of the food. To elaborate, pulsed electric field induces electroporation within the cell membrane, creating numerous hydrophilic pores when the charged plasma membrane exceeds the critical potential. The goal is to increase the number of pores on cell membrane to facilitate the irreversible ion and intracellular substance transfer out of the cell, which will undoubtedly lead to the destruction of cells. Given its mechanism, pulsed electric field are more popular in processing liquid and semi-finished goods due to higher conductivity.
For food preservation processes using various materials and technologies, it is crucial to review all the possible health, safety, and environmental implications. As such, Food and Material Safety Evaluation agencies such as FDA and EPA have released several guidelines on potential health risks associated with the use of nanomaterials in food. WHO has begun the Big Data Information approach to guide the food safety process decision making. Codex General Standard for Irradiated Foods is established in 2003 to recommend technological requirements for food irradiation. Even with the drastic advancement of various food preservation techniques, the challenges to the development of a healthy and sustainable food industry still remain. With the introduction and development of new technology in the food system, the public should also be well educated regarding the associated health, safety, and environmental implications.
In conclusion, food preservation is the critical control point to inhibit or eliminate microbial growth within food. It studies the influencing process and factors extensively to prolong or prevent the expiration and intoxication of food. A common practice involves the manipulation of microbial factors such as temperature, water activity, pH, and substance, as well as operating conditions. Given the challenges and complexity associated, food preservation is highly interdisciplinary that integrates the knowledge of machine learning, programming, advanced mathematics, biology, chemistry, and engineering to predict, explain, and control the population of microorganisms. As discussed within the report, current industrial methods that are widely adopted include thermal and cryogenic treatments, strategic storage, aerobic packaging, coating, dehydration, controlled distribution, etc., etc. Above all, food preservation is an imperative process for creating social and economic stability given its place in preventing foodborne illnesses. With that, it upholds the level of scrutiny discussed in the above section of the report and the impacts inflicted upon our society.